In this section, we will talk about how acidic and basic conditions affect living organisms. Now I'm going to talk about why the chemistry of life is sensitive to acidic and basic conditions. And I'm going to start by explaining that in an aqueous solution, a small percentage of molecules of water break apart into ions. So here we have the molecule of water, and it can divide into two very reactive ions. One that are the hydrogen ions that are positive, and the other one that are the hydroxide ions that are negative. And remember that both are very reactive. And so in a solution, if you have a lot of hydrogens, you will have an acid. And if you have a lot of hydroxide, it's because you have a base. And we will talk in the next exercise about that. As you watch in the video, remember that a compound that releases hydrogens to a solution is an acid. So, for example, this is the case. If you see here, you have a lot of much more hydrogens. However, in the opposite case, you will have a base where you have few hydrogens and a lot of hydroxide. Yeah, and in the case that you have an equal number of hydrogens and hydroxide, you will have a neutral solution. And how you measure that? You use a pH scale that describes how acidic or basic is a solution. The pH ranges from 0 to 14, and I know that you remember that from high school. And in 0 is the most acidic, so 0 will be this section, and the most basic will be 14. And I wanted you to stop the video for a second and try to look what are the biological compounds or the food that you found that like in which part of the pH scale they are located. Are more neutral, are more acidic, are more basic, and why? You can think about that. Okay, now that you thought about it, I'm sure that you saw the pure water. Living organisms contain a lot of water, so we are very close to neutral, and we have a lot of biological compounds that are very close to neutral, like human blood, tears, even seawater is very close to neutral. And on the other hand, more acidic, but also very close to neutral, are the urine and the saliva. But maybe things that we eat, like tomato juice, lemon juice, vinegar, are more acidic. And something that is very interesting is the gastric juice, that the cells in your body need to have a special adaptation to be able to digest the food, so they have to stand very acidic pH. In class, we will do some exercises related to this, and this is very important. Each pH unit represents a 10-fold change in the concentration. So if you go from a 7 to an 8 in pH, so the difference here is not one hydrogen of concentration, is a 10-time fold. Yeah, so here this one will have 10 times more, and this one 10 times less, and in 9 relative to 7, the pH 7, it will have 100 times less, and in 10 it will be 1,000. And we will do some exercises so you remember what you studied in high school. And if you don't study this in high school, go and check your textbook and do some exercises, because these kind of exercises will be in the test. But as I said before, we will solve some of these exercises in class. Now I'm going to move to another important topic that is very used in biology, especially in any lab. If you go to a biological lab, you need to know what is a buffer. And a buffer is a substance that minimizes changes in pH. And I'm going to show you a video of a buffer. And the idea is that a buffer will accept changes in pH and it will regulate those changes.
Here are two solutions, A and B, to which I've added universal indicator. Here's the color key. As you can see, both of these solutions are around pH 6, or very slightly acidic. Now, I'm going to add concentrated sodium hydroxide, which is a strong base, to each solution. It only took 1 drop of strong base to dramatically raise the pH of solution A. After 1 drop, the pH of solution B hasn't changed. Let's reset. Here are the same two starting solutions, A and B, with the same indicator. But this time I'm going to add concentrated hydrochloric acid, a strong acid, to each solution. It only took 1 drop of strong acid to dramatically lower the pH of solution A, but the pH of the other solution hasn't changed. Let's look at our pairs of solutions. When we added a strong acid or base to solution A, its pH changed dramatically after just 1 drop. When we added 1 drop of acid or base to solution B, its pH stayed the same. Let's add more acid and more base to solution B and see what happens. It takes much more acid or base to change the pH of this solution by the same amount. Now, how is solution B able to resist changes to its pH when strong acids or bases are added? How could we make and use such a solution?
So, for you to remember, a buffer will accept hydrogens when there is an excess, and the buffer will donate hydrogen when it's depleted. So it will allow to maintain the pH constant, what is very important not only in life, but when you are doing many experiments. Now I want to finish making a connection to what we learned, to something that is happening in our environment, the acid precipitation and the ocean acidification. And these two phenomena are putting on risk our environment, in particular the ocean ecosystems. You can study that in more detail in your textbook. You can read all this schematic in your textbook, but the main idea is generally, all the problems in our planet are because we are releasing too much CO2. Why? Because we are burning too much fossil fuels, and this CO2 is released in the atmosphere, and we're producing too much CO2, and this is producing many changes. In addition to climate change, it is also producing acid precipitation, and this is causing the reduction of the pH in the ocean. So we are changing all the chemical reactions that are happening in the ocean that you can see here. And this is having a lot of terrible consequences. One of them is that the CO2 is dissolving in the seawater and is changing the pH of the water. The oceans are becoming more acid, and the hydrogens are interacting more with our molecules, and those molecules, they were needed to the calcification of some marine animals, or for example to produce coral reef. So now those corals are going to die because they don't have the compounds that they need to survive. And you can read this in more detail in your textbook, but the main idea is, again, is that we have to be very careful with the amount of CO2 that we are releasing, because we are changing the balance of our planet.